Good evening, everybody, and welcome to the Macular Society's virtual clinic for June. Our virtual clinic webinars have become a very firm date in the calendar now and will remain so even when this interminable lockdown is finally over. And tonight's guest is a clear reason why these events are so incredibly valuable. It's a very great pleasure to introduce you to tonight to Dr. Gregory Hageman, who joins us this evening from Utah in the United States. Greg is the John A. Moran Presidential Professor of Ophthalmology and Visual Sciences at the University of Utah John Moran Eye Center. And he's also the Executive Director of the Steele Center for Translational Medicine. Greg is in fact a graduate of the University of Southern California, and he completed their undergraduate and graduate studies in biology and marine biology. His primary interest over the last 25 years has been directed towards the genetics and the origins of age-related macular degeneration. His research has been funded for many years by government, by charities and foundations, by industry, and is frequently, in fact, almost invariably conducted by with partners around the world. And he's a great collaborator, including with other scientists here in the UK. He is a prolific publisher of his research and an inventor of many new patents. Now, recently, he has founded a company called Voyant Biotherapeutics. This is a biotechnology company that is developing, among other things, therapies for the treatment of AMD, and has now he's now got funding for some early stage studies, phase 1 and phase 2 clinical trials that will test a new drug for AMD that have been developed, has been developed by Greg and his team. He is one of the most highly regarded researchers in this field anywhere in the world, and we are very, very fortunate to have him with us tonight. So Greg, a very warm welcome to the Macular Society Virtual Clinic. Thank you very much indeed for joining us. I'm going to hand over to you now for your talk. If you're listening to the talk, you want to ask questions, please use the chat function, and we'll have time for questions after Greg's talk. So Greg, welcome. Thank you so much for joining us. I'm going to hand over to you right now. Welcome.
Great. Thank you so much, Kathy. It's a real pleasure to be here with all of you this evening. I'm thrilled to be here. I enjoy this very much, and hopefully I can leave you with some take-home messages about efforts to develop therapeutics for macular degeneration. I hope your weather is much better than ours here in Salt Lake City. If I did my calculations correctly, we're about 43 degrees Celsius today, and so quite warm. But I'm excited to really talk to you about the efforts of the John Moran Eye Center and our teams. As I said, I thought I would start with kind of an overview of the presentation and some messages that I'd like you to take home with you after this presentation. So I'll give a short introduction to age-related macular degeneration, or AMD. I want to leave you with the idea that there are two major genes that are responsible for about 95% of all risk for developing the disease. Those two genes drive very different diseases. So in other words, AMD as we know it is really two very distinctly different biological diseases. And it's important that we understand the biologies that are being driven in those two specific diseases in order to gain the knowledge that we need to develop therapeutics that are really targeted specifically to chromosome 1 or chromosome 10, which are the two genes that I'll talk about quite a bit. After a short introduction, I'm going to focus the remainder of the talk on our efforts to develop drugs to treat chromosome 1 driven AMD. So I don't have to tell most of you in the room that, you know, AMD is clearly a major cause of what I would call irreversible vision loss in worldwide. The numbers are becoming staggering. I mean, there are recent predictions that about 196 million individuals are predilected for developing the disease. I don't have to tell you that it's truly a devastating disease for those of you that have macular degeneration. I was introduced to the disease about 35 years ago when I had a grandmother who developed macular degeneration. I was a marine biologist at the time, and really learned that the personal consequences of this disease, at least in my way of thinking, are completely unacceptable. And so that was the driving force for me to commit my career to studying macular disease. Kathy and I talked a little bit. I think in my way of thinking, there's an absolute need that we develop therapies to treat earlier stage disease. We'll talk about that a little bit. I think all the therapies we have currently treat late stages, and we really need to drive towards treating earlier stage disease. And I want to start, the term age-related macular degeneration, really age-related, as you all know, because the disease manifests typically in individuals over the age of 60. Macular degeneration refers to a degeneration of a very specialized region of the retina, which lies directly back in your visual axis. So that macula, or highly specialized region of the retina, is really specialized to drive your ability to see fine details, to read books, to drive cars. And it's that region that, for reasons that I'll explain, the macula is especially predilected for degeneration in this disease, which leads to, of course, loss of that fine acuity vision. So just a very short anatomy lesson. I'm going to talk quite a bit tonight about what's called the retinal pigmented epithelium. Think of it as a single layer of cells that lies back behind your neural retina and between the neural retina and the vascular supply for the back of the eye, which is called the choroid. So retinal pigment epithelium, RPE, is really the tissue that's taking the major insults in our studies, and I'll focus quite a bit on that. So I think all of you are familiar with, the most common line of thinking about this disease has been everybody has the same type of disease and everybody progresses in the same fashion. So typically we've talked about early stage disease, which is very much characterized by the deposition of these abnormal deposits called drusen back behind the retinal pigmented epithelium. And in about 50% of those early stage cases, those individuals will go on to develop one of two forms of late stage disease, either geographic atrophy or neovascular disease. And I hope by the end of this presentation, you'll come away with the idea that it's really not that simple. Again, two very distinctly different diseases with distinct biologies. So, I got very serious. We discovered the first gene that associated with macular degeneration back in 2000, published in 2005. And, the great thing about that was it accounted for about 50% of all risk for developing macular degeneration. So a single gene gave us a lot of hope that we could use that information to really develop therapies. If you kind of think about cancer, hundreds and hundreds of genes, so very difficult to find a common pathway to develop therapies. So I was invited by the chairman of the Moran about 14 years ago to move here to Utah and to set up a institute that was solely focused on understanding macular degeneration and developing therapies for it. We called that the Steele Center for Translational Medicine. And in the 14 years I've been here, we truly have enhanced our understanding of AMD. And as I think about that, I think the drivers of that have been, number one, people. The leadership here at the Moran is incredible. My team, which is comprised of about 40 individuals, scientists, clinicians, everything in between, is just really one of the most talented groups of people I've ever had the privilege to work with. We have tremendous supporters here in the state of Utah. The SCTM has raised somewhere over $40 million in the time that I've been here, largely because of those supporters. The second reason I think we understand the disease is access to resources, and by that we have about 60,000 study patients that we've worked with for many, many years. I realized 30 years ago that if we were to study a human disease, we really needed to study human tissue. And so I started a donor eye program 30 years ago. And that program by itself has really taught us a tremendous amount about macular degeneration. That resource is comprised now of about 12,000 pairs of eyes that have been donated. And as Kathy mentioned, the university, we created a partnership with Voyant Biotherapeutics back in 2011. And it's really Voyant that takes our discoveries and drives those towards therapeutic development. So let's go back in time a little bit. 35 years ago, I asked a very simple question, and that was, what are these abnormal deposits called drusen? What are they comprised of? And hoping that would teach us something about macular degeneration. It sounds like a simple question, but it took us years before we realized that drusen, and in fact the entire basal surface of the RPE, contains a lot of proteins that are associated with excessive activation of a very important part of your immune system called the complement system. So what's happening in those eyes, complement is being overactivated and it's being under controlled. And that results in the complement system doing what it's supposed to do, and it starts killing normal cells and tissues in the back of the eye. So it was really that initial discovery with complement that led us to the discovery of the first genetic association with AMD, and that was with a gene on chromosome 1 called complement factor H. And complement factor H regulates complement activation. So it turns out a year or so later, a second major genetic locus that associated with macular degeneration was discovered. That locus sits on chromosome 10 and contains a pair of genes called the ARMS2 HTRA1 gene complex. So two major chromosome loci, chromosome 1, chromosome 10, two major genes that sit in those locus with mutations, if you will. I think the important thing to point out is between those two genetic loci, we can account for 95% of all macular degeneration. So again, giving us great hope that we can use that to our advantage to develop therapies. So probably the first 5-8 years that I was here, we started taking what I think is a very unique approach to understand the contributions of these two genes in AMD. And the way we did that is we had enough donors and we had enough patients that we could take our donors and patients that only carried risk at chromosome 10 with no risk in the background at chromosome 1 and vice versa. We call those our pure chromosome 1 and 10 patients and donors, really to sort out, what pathways are chromosomes 1 and 10 driving irrespective of the other. And it's some of the most fascinating science I've ever been involved in. It didn't matter what we looked at, whether we looked in the eyes themselves at things like gene expression or clinical phenotype or histology, or whether we looked systemically, at blood cells or co-segregating diseases. Chromosome 1 and chromosome 10 drive two distinct biological diseases. And that's really the take home. Two very different diseases, one which is really complement driven, and chromosome 10 is really a vascular extracellular matrix driven disease. So just an example, ocular phenotypes are extremely different between these pure chromosome 1 and pure chromosome 10 patients. Things as simple as we just published a paper showing that retinal thickness the retinas were much thinner in the chromosome 10 patients, even before the manifestation of disease. Drusen and pigment epithelial deposits are really driven primarily by chromosome 1, and you rarely see drusen in chromosome 10 patients. Neovascular disease is much more highly associated with chromosome 10, whereas geographic atrophy much more highly associated with chromosome 1. So big differences in the way the disease manifests in the back of the eye and the way that it progresses. I put this slide in, so we have this wonderful database in Utah called the Utah Population Database that contains medical records, demographic records from all citizens in Utah, and this was created by the governor here some 70 years ago. So we have access to that, and we can ask questions like, clearly chromosomes 1 and 10 exist throughout your body, right? Those mutations exist everywhere. So I think it would be naive to think that they only cause macular degeneration. And indeed, we're learning that chromosome 1 and 10 drive very different systemic disease associations. In fact, they don't cluster at all. And the reason I mention this or bring it up is really the good side of this story is that any therapies we develop for chromosome 1 and 10 directed AMD should be efficacious in treating some of these other systemic diseases. So let me just summarize real quick the kind of the chromosome 1 and 10 directed AMD stories. Chromosome 1, as I said, patients with risk variants in chromosome 1 are not controlling the complement pathway. Complement is overactivating at this RPE choroid interface, so just on the basal side of the RPE, resulting in the death of the RPE. And the bad thing about that is the RPE does not have a capacity to replace itself. So once cells die, there's no capacity for replacement. Chromosome 10 affects the RPE as well, but we've shown now, and this paper will be published in PNAS very soon, that risk variants of HTRA1 result in decreased synthesis of HTRA1 protein specifically by the RPE and only in the RPE, again resulting in subsequent death of the RPE. So we believe very strongly that it's prudent to really think about therapies that are tailored to specifically target chromosome 1 and 10 pathways independent of each other. We've not been able to show that there are any common pathways shared between 1 and 10. Now, that's not to say that some individuals don't have both forms of AMD. And in fact, our patients who are risk at both 1 and 10 are the patients that very often develop AMD in their 40s and 50s. So I'm going to move on and specifically leave chromosome 10 out of the story and kind of focus on chromosome 1 as an example of how we're approaching therapeutic development. So chromosome 1, complement factor H, CFH, what I didn't tell you is back in our original discovery of that genetic association, I think I was most enthralled with the observation that there are risk forms of the gene that are highly associated with development of the AMD. But importantly, we discovered protective forms of the factor H gene. So, and these protective forms, if you carry 2 copies of protection, we all carry 2 copies of every gene. If you have 2 copies of risk, of course, you're at high risk for developing AMD. If you carry 2 protective copies, your likelihood of developing AMD is practically zero. The biology related to that is very extensive. We've actually been able to show that the protective form of factor H protein works much better at controlling complement activation than does the risk form of the protein. So now a very important comment that really has directed our therapeutic development. If you carry one risk copy of the factor H gene and one protective copy, you are highly protected. And what that says, based on what we know about the biology, is that all you need to do is put enough good protective protein back, and it takes over and controls complement even in the presence of some risk protein. So that really says that if we can get protective factor H back at the level of the RPE, that we should be able to reduce the odds ratio for developing the disease greatly. So a little bit about therapeutic considerations, drug development. I think a lot of pharma, especially large pharma, have not thought this through in many cases. But you really have to know what your drug target is. You know, what pathway are you trying to target? Where is that target, and what's its concentration? Once you know those things, you really need to think about what drug you're going to get back there and how you're going to deliver it. And I feel very strongly that we also need a very robust understanding of disease progression, especially in these pure chromosome 1 and chromosome 10 genotype groups. So the target, as I've said, the target is overactivation of the complement back behind the RPE, okay? And that complement in patients with risk is driving towards the formation of the piece of the complement system called the membrane attack complex that actually intercalates itself into cells and kills those cells. So lots of activated complement, lots of MAC at the level of the RPE in donors and patients with risk at chromosome 1. In individuals who carry 2 protective alleles, complement is very much well controlled and much less killing of RPE. And again, remember that the RPE cells, once dead, don't have a capacity to replace themselves. So the clinical consequences of all of that overactivation of complement, for those of you who can see this slide, it just shows a section through the macula of a donor who was homozygous protective on the top and a donor who was homozygous risk on the bottom. And essentially, as you start killing these RPE cells, the RPE detaches and it forms what we call pigment epithelial detachments, but think of them as just large blisters, and not really any different than blisters that you might get on your skin. So the RPE detaches, forms these large blisters, and clinically, very much like happens with a blister in the skin, as it gets too large, it bursts or pops. And that's exactly what's happening in the maculas of these chromosome 1 patients. These large detached regions or blisters they get so large that they can't sustain themselves and they collapse. And with that collapse, the neural retina underneath these large blisters begins to die without the support because there's no RPE to support it. So, and that results in the formation of geographic atrophy, which is very, very common in our chromosome 1 patients. We've spent a lot of time addressing the issue about concentrations of factor H that sit back behind the RPE, and the upshot of those studies is that we definitely believe, based on what we know about concentrations, that we can deliver appropriate levels of protective factor H back behind the retinal pigmented epithelium. So our overall strategy for chromosome 1 driven AMD is to tip the local balance of complement regulation. So let's put in a whole bunch of protective factor H back behind the RPE, and hopefully that protective factor H will do its job, regulate complement, and reduce this RPE cell death and associated pathology. So what that means is we have to get something back, factor H back behind the RPE, and our approach has been mainly to look at delivering protective factor H in a virus. So we take protective factor H, we stick that gene in a virus, we inject the virus into the region of the eye where we want that virus to infect, which is the RPE. Virus infects the RPE and begins to make a lot of protective factor H and secrete that into that sub-RPE space. So simple in theory. We've spent a lot of time figuring out how best to deliver protective factor H or these viruses to the RPE. Certainly in our hands, there's really three places we can inject, right? You can inject into the vitreous, which many of you have experienced with your anti-VEGF injections. Virus doesn't get terribly well to the RPE with that particular form of administration. We can put a needle between the retina and the RPE called a subretinal injection. That works very well, and suprachoroidally would be to inject the virus into that vascular bed called the choroid and have it then infect the RPE. So I think we'll end up probably running with suprachoroidal or subretinal injections in the future. And all of that work has been done in primates. We are getting just beautiful transduction and expression of protective factor H protein, in some cases with higher doses, maybe even too much, so we're backing down. But we are absolutely capable of delivering virus to the RPE and having it produce significant quantities of complement factor H. And so along those lines, we've just finished our large FDA primate toxicology study, and we're in the middle of analyzing those data as we speak. But I'm very confident that we will be able to deliver amounts of protective factor H. So the other thing I mentioned is we've had an ongoing natural history study, very, very important to understand in our pure chromosome 1 and pure chromosome 10 patients, the disease progression, if you will. Important to know that, and important to get away from individuals that carry, mixed genotypes, maybe one risk allele at 1 and 1 at 10. And so trials will most likely be conducted in these pure genetic groups. We've made great progress with the natural history study. We've spent a lot of time with Emily Chu using the ARADS cohort as a validation cohort. We're looking to submit our IND proposal to the FDA sometime in the next few months. And with all good luck, we expect to be in a phase 1, which is a safety study. So somewhere in the neighborhood of 12 to 18 patients, just really to make sure that there are no safety issues with respect to the therapy that we're delivering. And of course, if successful, that would move on to a phase 2 trial, hopefully sometime towards the end of 2022. So let me summarize here and again, leave you with some take-home messages. I think, good for us that 90% of all risk for developing macular degeneration can be explained by 2 genes. I think that's been a real gift to the field, that it's not 400 different genes. All of the data we've collected thus far really does suggest that AMD is 2 very distinctly different biological diseases. And again, you can have mixtures as an individual of those 2 diseases. Talked a lot about chromosome 1. Risk factor H does not regulate complement at the level of the RPE, leading to RPE cell death and dysfunction, followed by the clinical sequelae, which are the formation of these pigment epithelial detachments or blisters. We strongly believe that the expression of protective factor H at that interface should restore complement regulation in patients who carry the risk form of factor H. Preclinical studies in primates show us that we can deliver protective factor H to that RPE choroid interface using gene therapy approaches. And very much thinking along the same lines of development for chromosome 10. We're a little farther behind, but as of the last year, we very much understand what's happening with chromosome 10 disease. So I'd like to leave you with, please know that there's a whole team of individuals at the Center for Translational Medicine here at the Moran Eye Center, as well as all of our colleagues and collaborators around the world that are working very hard to attempt to alleviate this disease. And I truly believe we will win that day in the not so distant future. So thank you for taking the time to listen.
Greg, thank you very much indeed. What a fascinating, fascinating talk. Thank you so much. We've got some questions. I'll come to those in a minute, but just a couple of things on the key thing here that I think will have surprised a lot of people is that AMD is more than one disease. The patient groups and the pairs of the eye, the donated eyes tissue that you have is such an unusual, unique resource, isn't it? I think for the world, really.
Yeah, for the world it is.
And how difficult it is, we know from our own researchers and in the UK, how difficult it is to obtain human eye tissue. And this is so important, I think, because animals don't really have maculae, do they? So, I mean, lots of people object to research on animals, but in any event, they're not a great model for AMD, are they? Human eyes are really the only thing that's of huge use. Is that right?
Been my great belief that really that's the only model, right? We've certainly looked at so many of the animal models that just don't recapitulate anything like we see in the human tissue. So, and the other important part about the donors, we have tremendous eye donors here and their families, but we process all tissues within 4 hours of death, so I have a team of probably 8 people that are on call 24/7, and making sure that when we compare one pair of eyes to another, they're all processed within the same time frame, which becomes very important.
It's critical because I think I'm also given to understand that human eye tissue, retinal tissue, doesn't survive in a good state for very long post-mortem, does it?
Yeah.
I think that's right.
Very, very much so.
It's a very big challenge. It's a very big challenge to do this.
It is, and we are. The last I looked, we are getting about 1.5 donors per day. So it's a very robust program.
It is astonishing. So the other thing we know that, and we've reported, I think, to our members in the past, that there was great hope, and you touched on this in your talk about big pharma, major pharmaceutical industries, companies have put a huge amount of resource, haven't they, into looking at the complement, the impact of complement. And it was Roche, I think, who had a drug called lampalizumab that got into phase 3 trials, really late stage trials, when they must have spent hundreds and hundreds of millions of pounds on it, and it didn't work. So is this because they were not specific enough about which gene pathway they were actually targeting? What went wrong in that, do you think?
I think patient selection is a huge issue, correct? I mean, let's take Genentech with their factor D. It's a complement, an anti-complement drug, right? It might have, the sad thing to me is it might well have worked in patients who carried risk at complement factor H, but it's clearly not going to work in the other half of those cohorts, which are patients with chromosome 10. So silly to think that a complement inhibitor is also going to work in a chromosome 10 patient. So I think probably a lot of noise in those studies, and a lot of lack of understanding of biology. I mean, I think that if I the chats I've had with pharma, I had one the other day, we've shown that HTRA1 levels are decreased in patients with chromosome 10 risk. And drug companies, I know 5 that are developing inhibitors of HTRA1, which is only going to drive AMD to be even worse. So just bad understanding of the biology.
It's such a shame, because there was such a lot of hope, particularly people from geographic atrophy. Nothing on offer. It's so disappointing, isn't it, when such a late stage trial fails.
It is, and I truly believe we're on the right path, and I think a lot of others are falling in line. A lot of pharmaceutical companies are finally understanding that the genetics become important.
Could you say a little bit about the differences between the chromosome 1 and chromosome 10? So a question here is, do they manifest in different ways? Do people have different symptoms depending on which chromosome is involved, which gene is involved?
Very different. So chromosome 10 rarely associated with the formation or the deposition of drusen. So that was a big shock to me early on. We have no association between chromosome 10 and early stage grading, right, which is all based on drusen. So the chromosome 10 patients have a lot of vascular abnormalities in the retina, no drusen, much thinner retinas. They typically progress to late stage disease about 7, 8, 9 years earlier than the chromosome 10 patients. And it goes on and on like that. Chromosome 1, very much associated with lots of these pigment epithelial detachments, soft drusen, some people call them, in the macula, and the sequelae of events I described.
But both can be geographic atrophy or what we call wet
atrophy.
Except
it's in chromosome 1, I forget it's about 50% of those patients go to geographic atrophy and only about 20 to neovascular, but the neovascular phenotype is different. So the chromosome 1 largely drives what's called occult neovascularization, as compared to 10, which is very much driving classic and RAP lesions. So, we talk about CNV, and the field's just kind of forgotten that there are phenotypes of neovascular disease. And so those are very much driven specifically by 1 and 10.
Yeah. Okay, so there's some lots more questions coming in now. So just moving on to one or two other things that are coming in. So somebody's asked about statins, and I know there's quite a lot of interest in statins. What do you think the evidence or the potential is for statins? I mean, it's certainly not perhaps in this area, but are there other reasons why statins might be useful?
Well, I think the original thinking with statins was that, you do accumulate lipid, cholesterol in these abnormal drusen deposits. And I think that the original thinking was really, okay, if we can slow that down, we might decrease the progression rate of disease. And that's not borne out to be true. So I think statins really haven't been shown to slow down disease progression.
And amyloid, somebody else has raised the amyloid. There was quite a lot of interest in amyloid, wasn't there? And the as there is in lots of neurodegenerative conditions.
Yeah, drusen contain a lot of proteins that are quite similar to what you might see in Alzheimer's plaques, for example, or atherosclerotic plaques in large vessels. So they're there. It's, the attempts to target some of those have failed for reasons that, I think probably because chromosome 1 and 10 just are so much driving these diseases that doesn't mean that these other things might not exacerbate the effects of chromosome 1 and 10.
So just to go back, a couple of other people, just to clarify, on chromosome 1 and 10, it's not that one causes wet and one causes dry. They are slightly different versions that manifest in the same way. They look the same, but they've actually got different genetic origins.
Exactly, and then they're actually both killing the RPE, but by very different mechanisms.
Yeah.
And I think that's the important take home.
One of the things that I think distresses a lot of people, and I think certainly in the UK with lots of high street optometry practices now having OCT machines, quite a lot of people are being diagnosed with AMD who perhaps might not have been diagnosed in the past, when they're perhaps quite early on in the disease and maybe have a few drusen. It's quite distressing not knowing what the prognosis might be. I mean, you can't really tell, can you, who's going to progress at what speed at the moment, or even if they will progress.
Well, that's why I think diagnostics in the future will become much more important than we've allowed up to this point. Chromosome 10 is the good example. You don't really have a biomarker for 10 other than the genetics that says, you know, our 10 patients, in fact, many of those were called control patients when they were sent to us by clinicians because they didn't have drusen. And, you know, all of a sudden, no drusen, and they go straight to geographic atrophy or neovascular disease. So I think important to know your genotype. And I think diagnostics will turn around. And I think that's a really good example of why I think it's important.
Would a gene test actually be part of that diagnostic process? So are gene tests available now? Can people get gene tests?
You know, there are a couple small companies in the US that have been plugging along. There's a lot more discussion by the big pharma companies now, especially as we start stratifying trials based on genetics, I think. But, you know, there's 23andMe, there's Ancestry.com. The biggest problem, and I've talked a lot with 23andMe, is they don't account for protection at chromosome 1. And that's a big mistake, right? Because you might have one risk allele at chromosome 1, and 23andMe says you have an increased risk for developing the disease. If that second allele is protective, your risk goes way down. And so they're over-diagnosing potential risk for developing the disease.
That's not very helpful.
It's not helpful. No.
It's not very helpful. Is it even ethical to do that? I mean, I think if you can't.
It's kind of like these anti-HTRI1 trials, right? There's one in phase 2, and is it ethical now that we know that levels are already down to drive those levels down further? We're going to be faced with lots of questions like that.
I think there may have been a company in the UK that was private company that was considering offering gene tests. And I think they stopped in the end at that stage. This was 2 or 3 years ago, perhaps, because what do you do with the information? So the information that you give a patient who has a risk factor is the same that you give everybody anyway, which is don't smoke, keep your weight down, keep your blood pressure normal, eat your greens.
I think as we move towards tailored, if you will, tailored therapies, right, specifically targeted to chromosome 1 or chromosome 10, I think that's when you really do need a purpose, right? But I think the chromosome 10 example is, you know, I would want to know if I was 50 and I was homozygous risk at chromosome 10. You know, again, there's not much we can do about that today other than take vitamins and eat healthy, like you said, and stop smoking. But once therapies begin to come out, it'll be very important.
So I know that I have a science question here from a scientist who wants to know whether, in theory, we could find surrogate markers in OCTs, which could tell us whether a person had risky or protective versions of complement factor H and or HTRA1. So surrogate markers, this is, you know, you can't actually see the thing that you're looking for, but you've got something else that tells you whether it's there or not.
And we're all looking, we're looking a lot at OCT features, right? Especially in earlier stages prior to late stage disease. And I think there are lots of functional markers starting to come out that combined with OCT becomes very useful. Another good example is we're trying to figure out if we can detect it or not with OCT, but chromosome 10 drives the formation of these pathologies called basal laminar deposits that were worked on in the UK long time ago. those turn out to be driven by chromosome 10, and they result in a much thicker RPE Brooks membrane. So I think we'll be able to detect those. But again, I would argue that going back to the genetics first makes sense, and then following that up with surrogate markers.
Great. So just a couple of more general questions. A question here about ilea, aflibercept, which is an injectable drug for wet macular degeneration. And somebody's asking here about the average treatment duration, success rate, and what happens if it doesn't work. There may be a different process and protocols in the US, of course, but generally it's a pretty successful drug, I think, ilea, is it not?
I think successful for classic neovascularization and probably rapid, so more successful on the chromosome 10 front, I think. We struggle to get these anti-VEGFs back to the occult membranes. And I think, you know, again, neovascular disease is not neovascular disease. And I think it's important. But ilea has performed very well in those two types of CNV lesions.
And ranibizumab Lucentis a bit similar, that but it doesn't work so well in some people. In about 10% Lucentis, I think it doesn't work really at all. And this would be accounted for by the genetic differences that
I think so. I think so. I think we'll learn that, to be sure. And it may even direct, you know, which of the anti-VEGFs you use, or maybe even where they're injected. Maybe for occult membranes, you might be thinking about anti-VEGF into the choroid instead of into the vitreous.
And most people need this the anti-VEGF treatment of whatever form. Most people need it for life, don't they?
Yeah, and we're finding that the efficacy is falling apart, you know, somewhere around years 4, 5, 6. So folks are returning to kind of baseline at that point. But I would argue that that's a wonderful thing, that the fact that we can give somebody better vision for 5 or 6 years is a great thing. We just need to do better in the future.
There are longer lasting drugs coming through now, aren't there? That longer lasting anti-VEGFs, but I think anything for dry AMD is still quite a long way away, isn't it? I think a few years rather than
a few years. I think,
decades, perhaps.
Yeah, it becomes a political issue with FDA more than anything, right? We have to get past this endpoint of visual acuity. I mean, I think probably half of the audience can tell you that their visual acuity might be reasonable, but they're still struggling with their vision. And we have to figure out how to deal with that in earlier stage trials.
We've got some people on who don't have AMD, maybe have myopic macular degeneration. Is any of your work relevant to that?
Yeah, that's a tougher problem. I mean, I think, you know, there's been some progress in discovering genes that associate with myopia. Whether we can do something about those earlier on, I think is the question of the hour. I mean, what's really happening is similar to AMD. You're stretching that RPE more than it likes to be stretched, right? And it begins to fall apart.
Because of the shape of the eye, this is.
Because of the shape. Yeah. So very much the RPE again, but a different biology.
So possibly the chromosome 1, chromosome 10 thing isn't relevant, but as we found out that actually anti-VEGF treatment can be useful in people with myopic macular degeneration.
Yeah. So, no, it is for the neovascular form, for sure. Yeah. So.
On genetics, of course, if you have people will know that their family members, their mother had macular degeneration, their father had macular degeneration. The question here is my mother had macular degeneration. What are my chances of getting AMD myself? Is that possible to quantify yet from what you know?
Oh, absolutely. A genetic test will resolve that very quickly. In fact, we see a lot of the opposite, right? If you pick up a protective gene from one of your parents, you know, you pick up 2 protective genes. So, It's not a straightforward, mom had it, so I'm going to have it. And we've actually seen it go both ways. We've also seen the effects of chromosome 1 and 10 on top of other hereditary macular dystrophies, right? So retinitis pigmentosa, if you also are homozygous risk at 10, you're driving that retina to fall apart much earlier than you would say if you're an RP patient with protection in the background.
So another explanation for the difference that individuals can experience having been told they've got effectively the same disease, but they have wildly different experiences of how it plays out for them. It's bad luck, isn't it, to get both sets of
It's bad. I mean, and it's very common, well, not very common, but folks with Stargardt's and macular degeneration. But, we tend to focus on the Stargardt piece, but nobody thinks about these huge chromosome 1 and 10 genes in the background. So we're talking a lot with Julie Silvestri about looking in some of her macular dystrophy cohorts to look at the effects of 1 and 10 in the background.
Julie is a clinician researcher in Belfast, isn't she?
She is.
And a very good friend to the society, I must say. So in knowing that you have it in your family, determining the risk of yourself or your children is actually quite complicated, because you may have, although you may have the condition, you may have they may have another copy of the gene, which is a protective copy of the gene, which would reduce the risk from the other parent. So it's not directly hereditary, as some of the sort of single gene macular conditions can be, where you only need one copy of the faulty gene to be pretty certain of getting the disease. It's not that simple, is it?
It's not that simple, and it can go the other way, right? Both of your parents could have beautiful vision because they had one risk, one protective, and you ended up with one protective from each parent, and you're good. So
home and dry.
Yeah,
it's a
complicated. And we have that with our study patients all the time. We bring in a lot of families because they ask these questions.
We're getting near the end of our time. We've got a little personal question here, which is, how did you move from marine biology to ophthalmology? What has your marine biology studies brought to your work in AMD?
I guess biology is biology. It's just whether you do it in a fish or a human, I guess, makes the difference. It was a wonderful career. My first real job was to run the marine laboratory, USC, Southern Cal's marine laboratory on Catalina. So I did that for about 7 years, and owned about 2 pairs of shorts, and it was a very nice lifestyle. But really was struck when grandmother developed this, and she was fortunate. She developed it, late in life, but really struggled with it for the last 5 years, and it was a big lesson in my life.
I'm sure that's right. It's great that you've dedicated so much time to it. What's your big hope then for the future? What do you think the future is going to be? I know it's the $1 million question that researchers get asked, but what's the future look like, do you think?
I think it looks great. I think we'll have to start with later stage disease. I mean, just because of the FDA. But I think very quickly, Kathy, we will start moving back into earlier and earlier stage disease. I mean, the truth is we could fix these diseases the day you're born, right? I think technology will be an amazing thing. I've watched just in the last 10 years how powerful new technologies are, but the day will come where we can fix this disease very early, probably with some of the CRISPR technologies, but they'll be even better. And
gene editing technologies.
In the short term, though, I think we'll have some real successes here. We, as in the community, over the next 4 years for sure.
Well, Greg, thank you so very, very much indeed for giving up your time all the way from Salt Lake City to talk to us tonight. It's been an absolutely brilliant session, and I'm so grateful. So many compliments coming through on the chat.